Thank you for the introduction and thank you for the organization committee having me here. Good morning, everyone. I'm Summer. I'm a second-year PhD student from University of Pittsburgh, and now I'm working in Dr. Taylor's lab at the Drug Discovery Institute. I'm very happy to present our work today. So our goal is to develop a precision medicine platform using patient-derived iPSC liver microphysiology system for optimizing clinical trial design of non-alcoholic fatty liver disease. Yeah. Non-alcoholic fatty liver disease, or NAFLD, is a multifactorial metabolic disorder. Many factors contribute to the development of this disease, such as environmental factor, metabolic syndrome, demography, gut microbiome, and the genetic variants. Due to the complexity and the heterogeneity of this disease, NAFLD is recently proposed as metabolic associated fatty liver disease, MAFLD. So the starting point of our project is focusing on one of the most common variants that have been identified in the gene PNPLA3, as highlighted here, which I will talk about it more in my later slides. So now let's take a look of the disease progression of the NAFLD. So NAFLD usually started with the accumulation of the lipid droplets in the liver. The accumulative of the lipid toxicity will trigger the pro-inflammatory cytokine released from the immune cell. And it will further lead to the activation of the astrocytes for the fibrosis genesis. So the disease will progress from the early stage of NAFLD to an advanced stage NASH and to a later stage cirrhosis. So a good disease model should be able to recapitulate the disease progression, as I mentioned here, and as well as the key disease phenotypes. So as you can see from this map, NAFLD is a very common disease globally. And the high-risk variant of PNPLA3 is also a very common variant. And you may also notice here, You may also notice here, this high-risk allele is more common in certain population with a higher prevalence of NAFLD, such as the Hispanic population or the Latino population. Thus, the prevalence of this PNPLA3 high-risk variants accounts for a large fraction of the inter-ethnic variability in the NAFLD susceptibility. So our initial strategy to address the heterogeneity of the NAFLD is to stratify the patient population based on their genetic variants. So in this case, it's this PNPLA3 high-risk variant. So we will group the patients by the genetic variants for a more effective and more targeted treatment. So far, we have recruited more than 200 patients through UPMC Flow Clinic, and we have genotype and identified different genetic variants from our patient cohorts. So you can see all the variants listed here has been identified in the GWAS study showing association with the NAFLD. And they have a range of different effects. And PNPLA3 high-risk variants is the major one with, is the most common one with a major effect. And you can see this is a C to G mutation. And the C is the major polymorphism and G is the minor one. And you can see from on the right, we compare the genotype frequency to the different disease condition. We have the healthy individuals, we have the NASH group, we have the end stage of the liver disease here. And the minor homozygotes are showing in the yellow box, and the heterozygotes and the major homozygotes are in pink and purple respectively. And you can see compared to the other variants, the genotype frequency of PNPLA3 is increased along with the disease progression. And very interestingly, on the very right, you can see the prevalence, the presence of this polymorphism is not necessarily related to the development of type 2 diabetes. Now you may be wondering, what is the biological function of PNPLA3? PNPLA3 encoded a protein that is highly associated with lipid metabolism. In the normal condition, this protein is located on the lipid droplets, and it functioned as a hydrolase to facilitate the metabolization of triglyceride and the fatty acid in the hepatocytes and the retinol in the stellar cells. However, the mutant form of this protein will block this process, which lead to the lipid accumulation in different cell types. So we used human primary hepatocytes to examine the intracellular lipid content with or without this mutant. And we used NAFLD to detect the lipid content. NAFLD is a dual probe. It will turn fluorescent green in a lipid-rich environment. Otherwise, it will stay in red. So you can see from the bottom image, which is the mutant hepatocytes, it shows a significantly higher intracellular lipid content. And the quantification here indicated that the normal human hepatocytes carrying this PNPLA3 high-risk variants present significantly higher content of intracellular lipids. But as I mentioned before, NAFLD is a complex disease resulted from the interaction of different cell types. That's where our model come into the story. We have built the lipid acinus microphysiology system, or LAMPS, to recapitulate the major structure of function, major structure and function of human liver. The liver acinus is the functional units of the liver, which consists hepatocytes, Kupffer cells, stellar cells, and the sinusoidal endothelial cells, and the cholangiocytes. The blood flow in it create the oxygen gradient zonation. Based on this physiological structure, we build our LAMPS model. LAMPS is a 3D microfluid device. The primary hepatocytes co-cultured with the key non-parenchymal cells, as I mentioned here, and we will culture them for 10 days with a flow exposure. It can be ran in either zone 1 or zone 3 oxygen level. To recreate the physiological and the pathological environment, we designed 3 medias, starting with the normal fasting here. Starting with the normal fasting here. The normal fasting media is to recreate to mimic healthy liver condition with a normal level of glucose and insulin. And then we are able to drive the disease to the early metabolic syndrome, EMS, with increasing level of glucose, insulin, and the addition of free fatty acid. And next, we're able to drive the disease to late metabolic syndrome, LMS or NASH, with the immune stimuli such as LPS and TGF-beta. We also develop a matrix to quantify the disease-related phenotypes such as albumin secretion, hepatic steatosis, oxidative stress, inflammatory cytokine release, stellar cell activation, fibrosis, and etc. And all the measurements here are consistent with the clinical data. Sorry, it's frozen. Okay, yeah. To build a patient-specific liver MPS system required the collaboration from clinical side to the bench side. This is a multi-PI work funded by NIH. We have Dr. Bihari. Dr. Bihari from the UPMC Flow Clinic. We have Dr. Suto Gutierrez from the pathological department at the University of Pittsburgh. And we have Dr. Taylor's team from the Drug Discovery Institute at the University of Pittsburgh. So in our first aim, we will construct the liver MPS system using primary liver cells with or without this PNPLA3 mutant. We will drive the disease progression in our model. And the data gained from here will serve as the fully adult reference for the later iPSC-based liver MPS. And we will build our model using iPS, using patient-derived iPSC cells, and the result we will compare the results to the first aim by comparing to the primary-based liver MPS. We were able to answer the question, to what degree this iPSC-based liver MPS is able to recapitulate the adult liver function and the disease phenotype. Okay, in the end, we are going to use our patient-specific liver MPS for clinical trial optimization. Regarding of the primary cell-based liver MPS, currently we are able to evaluate our first set of human primary cells with or without the PNPLA3 mutant. So as you can see from here, we examine the steatosis phenotype by quantifying the intracellular lipid content. As shown in the graph, compared to the wild-type model in the blue, the mutant model in the red show a significantly higher intracellular lipid accumulation in the disease-induced environment, showing in here EMS and LMS. In addition to the intracellular lipid content, we also measure the secreted triglyceride. As you can see here, we compare the wild-type model and the mutant model. We see a significantly higher increase of the secreted triglyceride level in the late metabolic syndrome media. So taken together, our preliminary results indicating that this PNPLA3 mutant may introduce a burden in a disease-induced condition, and it may further accelerate the disease progression. Now we are working on more lots of the human primary hepatocytes to confirm our initial results here, and more characterization matrix are carrying on to evaluate the disease phenotypes such as fibrosis or cytokine release. Oh, sorry. Is it frozen again? Oh, okay, sorry. Yeah, regarding of the iPSC-based liver MPS, we are going to generate the liver, the iPS-derived liver cells from patient PBMC sample. In our initial step, we will integrate iPSC-derived hepatocytes, stellar cells, vascular endothelial cells, and the cholangiocytes into our liver microphysiology system. In the future, we will also integrate the iPS-derived immune cells in our model, such as macrophage cells. The iPS-based liver microphysiology system is still at a very early stage. We have tested a range of different conditions and parameters and developed an optimization protocol to maintain the iPS-derived hepatocyte viability and the maturity in the LAMPS model after 8 days with flow exposure. As you can see here, the IHABs co-cultured with non-parenchyma cells in the LAMPS model for 8 days with flow exposure. And the image showing here are represent the area of interest on our chip. And the GFP expressing IHABs found a nice and homogeneous distribution on the chip. And at the bottom image, you can see the red signal is HNF4 alpha, which represent the adult hepatocyte marker, indicating the maturity of the IHABs. we are able to maintain about 70% of the maturity of our IHABs in the LAMP model for 8 days flow exposure. Meanwhile, we also developed disease media to induce the fatty liver in our model, and the fatty liver disease is induced by the addition of free fatty acid. Compared to the control group, we see a significantly increased of the lipid content here. We have DAPI here showing the nuclei, and the red signal is from lipid staining. And from the quantification, we can see the lipid accumulation is threefold higher in the disease-induced media with the similar nuclear with the similar cell amounts showing the nuclear count. And in this study, we use biosystic platform to establish and evaluate our reproducibility of our model. And this platform is very useful. It provides tools for us to evaluate and review our experiments with different settings. So our iPS-based model is still in development stage. So here is showing one example that how we use this platform. And the lines here you can see is from three different IHABs batch, and we collect all the data and put into the database. And you can see you can see highlight here our reproducibility is getting better and better. And now is the summary of my talk. We have recruited and genotyped more than 200 patients for this study, and we have established the protocols for the major liver acinus iPS-derived cell types. We have evaluated the first set of human PNPLA3 low-risk and high-risk hepatocytes in the LAMP model and found differences in the intracellular lipid content and triglyceride released between them. We're working on more measurements, including fibrosis and cytokine release panel. iPS-derived hepatocytes have been demonstrated to maintain the phenotypic integrity and the maturity on the flow for at least 8 days. So all these results have paved the way toward our final goal, to optimize clinical trials using patient-specific liver MPS system. So last but not least, I would like to thank our wonderful group and the great collaboration, and also thank NIH for the financial support. And that's all my talk today, and any questions or comments are welcome. Thank you.
Thank you, Summer. Wonderful talk. We have time for one question. Okay, I will take it then. In the projects that you are involved in, you have a couple of aims. Aim 1, where you assess the disease phenotype in the primary cells, and in aim 2, you're actually generating iPSCs. Are those iPSCs coming from the blood of the patients from the A1?
For M1, we are using the cryopreserved primary hepatocytes. In the patient-derived iPSC in the M2, we will using the patient that we recruit in the clinical trials that I mentioned.
Yeah. Those going to be different patients.
Different patients.
Not this 200. Okay.
Yeah.
Thank you very much.
Thank you.